Assalamualaikum and hi, my name is Asmar.mp4. Today I would like to present about my final year research project which on title Antibiogram of Salmonella species isolated from Malaysian giant freshwater prawn Microbacterium reservigilava. Giant freshwater prawns have become a major species of aquaculture and are rapidly growing in many countries. It is also becoming an increasingly important target species in Malaysia as its culture is regarded as having the potential to increase income among poor farmers. Salmonella species is one of the main source of foodborne sicknesses, and there is currently no database of antibiogram available for bacteria from the giant freshwater prawn hatchery in Malaysia. For most countries, aquaculture is one of the key economic tools and it is one of the essential components of food security. The aquaculture industry is rapidly and intensively growing in Malaysia as well. And in the aquaculture industry, disease is a major cause of problem because it can cause high mortality and cause tremendous losses to farmers and the industry at the same time. For the null hypothesis, can isolate and identify bacteria Salmonella species from Malaysian giant freshwater prawn Microbacterium reservigilava rearing tank. Secondly, can characterize antibiogram of bacteria Salmonella species from Malaysian giant freshwater prawn Microbacterium reservigilava rearing tank against 20 types of antibiotics, while the alternative hypothesis is vice versa from the null hypothesis. These are the objectives. Microbacterium rosenbergi or giant freshwater prawn, locally known as udang galah. It is found all through the tropical and subtropical territories of the Indo-Pacific area from India to Southeast Asia and Northern Australia. It is one of the greatest freshwater prawns on the planet and broadly developed in a few nations for food. Salmonella is a genus of rod-shaped bacillus gram-negative bacteria of the family Enterobacteriaceae. Two types of Salmonella which are Salmonella enterica and Salmonella bomori. Salmonella was named after Daniel Elmer Salmon, an American veterinary specialist. Gram staining is a type of staining used to identify and classify bacterial organisms into two broad groups. Firstly, gram-positive bacteria and secondly, gram-negative bacteria. It is also known as the Gram method and the name comes from Hans Christian Gram, the Danish bacteriologist who invented in the technique. The DIS diffusion test or agar diffusion test or QB-Bow test is an antibiotic susceptibility test. It uses antibiotic DIS to test the extent to which bacteria are affected by those antibiotics. In this test, wafers containing antibiotic are placed on an agar plate where bacteria have been placed and the plate is left to incubate. For sampling, the parent stock of giant freshwater prawn was brought from Perak. Only the buried females were taken. The giant freshwater prawns were transported in a plastic container with ample aeration. The giant freshwater prawns were then acclimatized. Formalin was added into the water of 10 ppt salinity in order to kill any harmful bacteria or parasites. After a whole night, the water was changed and the larvae started to hatch. For isolation serodilution, a series of 10 test tubes containing 10 ml of sterile distilled water were prepared. By using a sterile pipette, 1 ml of the water sample from giant freshwater prawn rearing tank was added in the first tube of the set. The test tube was labeled as 10 to the power of -1. The contents were well mixed by swirling the tube upside and down for a few times, and from the first tube, 1 ml of the sample was taken and transferred to the second tube, and the test tube then was labeled as 10 to the power of -2. The procedure was repeated with the remaining tubes, labeling them until 10 to the power of -10. These are the methodology for isolation spread plate. For identification gram staining, a smear was prepared. The smear was stained with crystal violet for 1 minute and then rinsed with distilled water. The smear was stained with grams iodine for 1 minute and then was rinsed with acetone. The smear was rinsed with acetone until the blue color stops coming out of the smear, and then the smear was rinsed with distilled water again. The smear was countertained with safranin for at least 10 seconds, and after 10 seconds, the smear was rinsed again with distilled water and was air dried. The slide was completely dry before put under the microscope. Bacteria suspension stocking bacteria. A single colony was chosen and subcultured into a beetle bottle by TSB. The mouth of the beetle bottle was sterilized by flaming it near to the Bunsen burner. By using a sterilized loop, an isolated colony was taken from agar plate and transferred into the TSB beetle bottle. The loop was shaken in the TSB until the colony seems to fall in it. The mouth of the beetle bottle was sterilized again by flaming near to the Bunsen burner, and the procedure was repeated for the next 9 bottles which were incubated overnight. This diffusion method, the stock culture from bacterial suspension were observed after leaving overnight in the oven The liquid of the stock culture was turned cloudy. A cotton swab was dipped into the bijo bottle with stock culture for 1 minute. The same cotton swab was then sprayed on the surface of the TSA plate. After that, a sterilized forceps was used to pick up antibiotic discs. 6 to 7 types of antibiotic had to be placed on the surface of each agar plate. The antibiotic used are as shown. As shown in the figure below, with specimen under 100 times magnification, negative gram stain and the medium-sized bacillus was the Salmonella. Salmonella bacteria will appear as pink rods when viewed under the microscope. This suggests that it is a bacterium that is gram-negative. In bacteriology, the gram stain is the most commonly used staining technique. With the gram staining technique, bacteria that stain purple are considered gram-positive. Those that stain pink are said to be gram-negative. As well as preventing osmotic lysis, the bacterial cell wall helps to give the organism its size and shape. In both cold-blooded and warm-blooded animals, as well as the climate, Salmonella is a genus rose-shaped bacteria occurring worldwide. Salmonella strains cause diseases such as typhoid fever, paratyphoid fever, and food poisoning. In this study, a total of 32 isolates of Salmonella species were successfully isolated from water sample, which were observed on the 30 agar plates. Salmonella species appears to be yellowish on agar. The total plate count of Salmonella species from water sample ranged from 0.01 to 0.6 colony forming unit. In the present study, the majority, more than 80% of the bacterial isolates present were found to be resistant. However, the percentage of bacterial isolates present which were sensitive ranged from 0% to 10%. Furthermore, the percentage of bacterial isolate present which were intermediate sensitive also ranged from 0 to 10%. Overall, the sensitive category accounted for 80%, where 10% and 10% were reported as sensitive and intermediary sensitive categories. These are the graph and the table of antibiotic susceptibility of Salmonella species isolated from giant freshwater prawn Microbacterium rosenbergi. Antibiotic agents can be used as a tool to maintain health and disease prevention. Overuse of these antimicrobial agents can potentially result in antibiotic-resistant incident in pathogenic bacteria, making them less responsive to antibiotic. Isolation and identification of the bacteria is a qualitative test which study the culture characteristic of bacteria on the selective media such as GSP agar. Bacteria can enter M. rosenbergi hatchery system through feed, broodstock, and rearing water. Since the enormous amount of Salmonella species were discovered to be available in the water test in the M. rosenbergi hatchery, hence we may presume that these bacteria were begun from water source of the hatchery. Water source may contain high organic materials that added to the presence of big amount of Salmonella. In this study, approximately 80% of the total isolated bacteria were resistant to the most of the antibiotic used. On the other hand, 90% of the bacterial isolates obtained from M. rosenbergi hatchery in India were immune to furazolidine in a study by Sahul Hamid. Therefore, compared to the Indian scenario, furazolidine is still effective in combating the bacterial diseases of giant water prawns in Malaysia. Sahul Hamid also clarified that the occurrence of high-resistant furazolidine bacteria in the Indian hatchery was due to extensive prophylactic use of this antibiotic. As a result, Sahul Hamid advised that antibiotics should not be used for prophylactic purposes except for giant freshwater prawn hatchery treatment. In this research, bacterial isolates were immune to most antibiotics. This may be due to extremely antibiotic-contaminated water supplies in the hatchery. Since this hatchery did not use antibiotic for treatment and prophylactic use, it is likely that the antibiotics that reach the hatchery's water supply may come from nearby industrial waste or agricultural activities. The presence of a high incidence of resistance to most of the antibiotics tested indicates a rise in seawater antibiotic contamination. Media also reported a similar finding in which gram-negative bacteria were isolated from seawater, soil, and shrimp in Iskandarian Bay, Turkey, showing high resistance to the antibiotics tested. The authors reported that in the Bay, Turkey, the incidence of bacteria highly resistant to the antibiotic tested was due to industrial contamination. However, the contribution of bacterial ecology to the incidence of antibiotics among bacteria through mutation, selection, and the flow of genetic information between bacteria cannot be denied. To conclude, findings from this study demonstrated that Salmonella species isolated from giant freshwater prawn were multi-resistant to antibiotics. Excessive input of antibiotics may favor for the development of co-selection and cross-resistance in the bacterial community of the aquaculture environment. Hence, aquaculture experts or farmers must be prudent in practicing health management and may consider natural products and vaccine as the alternative measures. It is recommended that shrimp farmers may use these antibiotics in shrimp or prawn culture for prophylactic and treatment purposes. However, all strains of salmonella species cannot be destroyed by this antibiotic. Further studies should therefore be carried out to find out which antibiotic can inhibit the growth of salmonella species strains. Instead of searching for other successful antibiotic, we propose that antimicrobial agent extracted from natural resources could be used by aquaculture experts. That's all, and thank you for watching.